In the last lab, we looked at the macromolecules that make up the major components of our cells. But now let's look a little bit larger. Let's look at the cell itself and the various structures and functions that these cells have and do throughout their regular daily lives. So when we're looking at cells, we're looking at the smallest classifiable unit of life. And that these organisms, living things we're looking at, are made up of at least one cell. This is part of what we call cell theory, that the cell is the basic unit of life, that all organisms are made of at least one cell, and that cells can come from, or have to come from, I should say, from pre-existing cells. Some organisms are one cell and we call that unicellular, but since this is a human biology class, we're focused on the components or the organisms made of many cells that are multicellular. So us as humans are multicellular organisms. And just like our body has different components, right? A heart, a liver, a stomach, a pancreas. Just like that, our cells have smaller components inside of them, and instead of organs, we call those organelles. So let's look at a model of the kind of generic animal cell and some of the components. And there's a lot of components to it, so we're going to look at just a handful. Let's start down here at the bottom where we have the plasma membrane. So the plasma membrane is going to kind of help keep everything all together. And we'll get into more detail on each of the functions of these, but let's just kind of get a visual first. So the plasma membrane is this kind of sac-like container or a structure that's holding everything in. We have a peroxisome, and this can help with detoxifying things. We have ribosomes. These little tiny dots you see there are ribosomes. We have the nucleus, which is this large purple piece, and within it we have a nucleolus, a nuclear envelope, and nuclear pores. Ribosomes are also kind of hanging out with it. We have a smooth endoplasmic reticulum, also known as a smooth ER, and a rough endoplasmic reticulum, also known as the rough ER. The difference between smooth and rough comes from their appearance. The rough ER looks rough because it has ribosomes embedded in it, so it has a studded appearance, whereas the smooth ER does not have any ribosomes, and so it has a very smooth appearance. We'll talk about how that changes the functions of those two in a little bit. We have the cytoskeleton, which is composed of microtubules, filaments, intermediate filaments, and actin filaments. The centrioles, which are going to help kind of organize parts of the cytoskeleton. The cytoplasm, or the cytosol, which is the fluid. A mitochondrion. A secretory vesicle. Lysosomes, and the Golgi complex, also known as the Golgi apparatus. So you'll want to be able to identify these, not just in terms of their names and functions, but also in how they look. Get familiar with images of animal cell models so that you could identify the smooth ER from the rough ER, the Golgi from a lysosome, the centrioles from a mitochondrion. Let's get into their functions. So that nucleus, that big purple sphere we kind of saw hanging out in the middle, is really kind of like the control center for our cell, and it contains DNA. This is where all of the genetic material is contained and is organized. So you kind of think of it like the brain, so to speak, of our cell. The plasma membrane is our containing unit, or our containing portion, that keeps things inside the cell. And it can help regulate what comes in or leaves out of the cell. Vesicles are kind of modified parts of the plasma membrane that can transport materials both in and out of the cell as well as through the cell. So if something needs to move from the smooth ER to the Golgi apparatus, a vesicle can transport it. It kind of functions like a box or a moving van, so to speak. The smooth ER produces lipids and carbohydrates. It can also perform very specialized tasks like certain ion storages, and that can vary depending on what type of cell we're looking at. So later on, to give you an idea of what we're talking about, When you look at the muscular system, you'll talk about the smooth ER again, and one of the specialized tasks it provides for muscle cells in particular. The rough ER, which has ribosomes with it, is going to work to produce proteins. The ribosomes are really the workhorse pieces that are making the proteins, but the rough ER is helping with that and also kind of processing and editing things as needed. Both the smooth ER and the rough ER are going to send components to the Golgi apparatus. The Golgi apparatus is going to package, process, and transport those materials around. So together we call this the endomembrane system of the smooth ER, which is making our lipids, the rough ER, which is helping produce proteins, and the Golgi apparatus, which is packaging and processing and moving things around. I like to call the Golgi apparatus the Amazon fulfillment center of the cell because Amazon does not make the things that you need. It just packages them and assembles things together for you and ships it to your house. It's kind of what the Golgi apparatus does. The last few we're going to look at in detail are lysosomes, mitochondria, and the cytoskeleton. Lysosomes use digestive enzymes to break down foreign material or old cell parts. So basically, it's like a recycling factory, so to speak. If there's some sort of molecule that needs to be broken down, or there's like an old cell organelle that needs to be broken down, or let's say we're a white blood cell and we've taken in an invading bacteria, we can use these digestive enzymes to break these things down and render them harmless. Mitochondria, if you're familiar with the internet, you know, is the powerhouse of the cell. And while that's a good start to understanding what the mitochondria do, it's not quite enough. When we say powerhouse of the cell, that's really generic and it doesn't really tell us what we're doing. We got to go deeper. The mitochondria are using a process we call cellular respiration to produce a molecule called ATP, or adenosine triphosphate. Adenosine triphosphate is our energy currency within the body. ATP is going to help power a lot of chemical reactions within the cell and in other cells too. So we need to produce a lot of ATP to help make sure that all of these reactions we need to get done have power, so to speak. The last one we're going to go over is the cytoskeleton. This gives us some structure and also helps with movement. So instead of just being a flat blob, just kind of a mess, we have a way to kind of hold ourselves up, we have a way to kind of hold ourselves out, and if we need to move around through things like cilia or flagella, the cytoskeleton and the filaments that are part of the cytoskeleton can help provide that movement. Speaking of movement, as cells, we need to move things in and out on a very regular basis. But there's a couple ways that we can move things around: diffusion, facilitated transport, and active transport. Now, both diffusion and facilitated transport are passive processes, meaning we don't need to use any energy, ATP, in order for these things to happen. They're going to happen relatively naturally. Diffusion is the random movement of molecules from a high concentration to a low concentration. So if we have a high concentration of sodium on one side of a membrane and it passes through using diffusion, then the high concentration would move towards the low concentration until we reach equal concentrations on both sides. Facilitated transport is the same idea, that we're moving molecules from a high concentration to a low concentration, but for whatever reason, the molecule or substance is not able to move freely through the membrane, and instead we have to use transport proteins, some sort of channel, some sort of protein that can help alleviate that or provide a pore or an opening for that molecule to get through. Active processes go against that gradient, meaning we're moving something from a low concentration to a high concentration. Now, the low to high concentration movement is not going to happen naturally. We have to put in some energy, ATP, in order to force this transport to happen. So diffusion and facilitated transport are free, essentially, to do and move from high concentrations to low concentrations, whereas active transport, we're moving from a low concentration to high concentration, and we're going to have to pay for it to happen. Osmosis is a special type of movement where we're focusing on how water moves across a membrane. In order to understand how or why water is going to move across a membrane, we have to understand tonicity, which is the relative concentration of solutes and the solvent. In this case, the solvent would be water. And there's a few terms we can define: isotonic, hypotonic, and hypertonic. So let's look at tonicity and some of our cells, namely blood cells. So hypertonic, isotonic, and hypotonic. With isotonic solutions, and again, when we're using these terms, we're describing the environment, not the cell. We're talking about the liquid the cells are hanging out in instead. In an isotonic solution, the solute concentration is about equal, both inside the cell and outside the cell. So the environment we put our red blood cells in is about the same as the environment that is inside of the cells. So as water moves in, water also moves out. It's a net zero, so to speak. With hypertonic solutions, we've put our cells in something with a really high concentration of solutes. Water is going to move out of the cell into the high concentration of solutes. Think of it like the water's trying to dilute down the high concentration of solutes. Because of that, water leaves the cell, but it does not go back in, giving the cells a shriveled appearance. We call this crenation. In a hypotonic environment, we have very little to no solutes in the environment, meaning the high concentration of solutes is found inside of the cell. Well, remember what we talked about with the hypertonic solution is that water wants to dilute the high solute concentration. So if the high solute concentration is inside of the cell, water will flood in. Because so much water is flooding in and not leaving, we get a puffy, kind of poofed up appearance where we've kind of been blown up like a water balloon. And sometimes we can be pushed past the point of survival and the membrane will actually give way and burst open. We call that lysis, when the cell breaks open. So if you were to put something like a red blood cell in pure water, which has no solutes whatsoever, water would rush into the red blood cells and they would all rupture, killing the cell and rendering them into teeny tiny little pieces. There's also enzymes that we mentioned before that are special types of proteins that can help speed up reactions inside the body. Enzymes are like other proteins in that they have a very specific range that they can work in. So most proteins have to have a specific temperature range, pH range, so on and so forth. If for whatever reason it gets too hot, too cold, too acidic, too basic, whatever, then the enzyme won't necessarily work. Catalase is one of those enzymes that helps reduce things like hydrogen peroxide to water and oxygen. Enzymes are going to come back a lot in this class, so you'll want to pay attention to what an enzyme is, what they do, and what type of environment they have to work in. These are one of the main reasons why we have to maintain homeostasis, that generally constant environment inside of our bodies, because if we didn't, these enzymes that we rely on would not necessarily work.